Hello everyone, I'm Steven and you're watching Steven Loves Science. Now in today's video we are going to hopefully gain an intermediate level understanding of the process of mitosis, or the process in which a eukaryotic cell is able to produce two new identical daughter cells. So for those of you familiar with the cell cycle, mitosis is going to begin after the completion of interphase. So the G1, S, and G2 phases have already been completed. And now if we look at the next frame, we can see the result of that. So the DNA has been replicated, forming two identical sister chromatids joined at the centromere. Right now we're really diving into the first stage of mitosis, which is prophase. So as you can see, we have these very defined compact structures, which are the pairs of sister chromatids. And we can also see that the nuclear membrane has kind of become dissociated into these small vesicles, and those small vesicles will eventually disappear. This is important because it will allow the sister chromatids to be accessed by the mitotic spindle, which at this point is already beginning to form. We are now entering prometaphase. In this phase, the spindle fibers are going to begin to interact and attach themselves to the connect core of the sister chromatids. So by the end of prometaphase, the mitotic spindle is completely formed. Eventually, the pairs of sister chromatids align themselves along a plate called the metaphase plate. So at this point, we are now in metaphase. So each pair of chromatids is attached to both poles by the connect core microtubules. And as you can see, they are aligned into a single row so that each daughter cell will receive one copy of each chromosome. So at anaphase, which is the phase we're in now, the connection responsible for holding the chromatids together is broken, and each chromatid is now an individual chromosome. And as anaphase proceeds, each chromosome is brought closer to the pole of which it is attached to, and this just involves the shortening of the connect core microtubules. And in addition, the poles themselves move farther apart due to the elongation of the polar microtubules due to the action of motor proteins. Now during telophase, the chromosomes reach their respective poles and begin to decondense. The nuclear membrane now begins to reform to produce two separate nuclei. We have now reached the last stage of mitosis, which involves the separation of the two new daughter cells. At this stage, which is known as cytokinesis, a contractile ring composed of the motor proteins actin and myosin forms. Now myosin then hydrolyzes ATP, which shortens the ring and subsequently forms what is known as a cleavage furrow. This cleavage furrow ingresses on itself until the two cells are finally separated. So that's about it for mitosis. And if you want to check it out, I actually wrote a poem on mitosis, which is available in the description. So as always, take care and thanks for watching.